pleasure today to introduce an old friend, a colleague. Some of you, probably most of you already know John. John was here not even 10 years ago. Went over to California, but they're all going over there. So it's good to bring him back here where all the excitement is. John, as you know, trained with John with Shostak, Josh Shostak, years ago, and has been, playing around in this kind of colorful space of synthetic biology, kind of how to create things that might work on their own, but are not part of our natural phenomenon. So it's really interesting to hear more about what he's been up to. I think Google's been interested in him. He's been doing some stuff with them in recent years. So I'm not going to belabor this anymore, but introduce John to talk about some of the latest stuff he's up to.
Thank you very much. I want to begin by thanking Josh for the invitation to speak here today. This is just a really wonderful opportunity. It feels like a reunion of sorts. It's a great chance to sort of reunite with old friends and certainly make new friends. So today, kind of what I want to tell you about is some of the things that we're doing in my lab at UCI. And my lab very much works at the interface of, say, synthetic biology and what might be considered early stage drug discovery. And so folks that are working in that field are kind of asking two primary questions. The first question is, what are the drugs in the future going to look like? And the second question that goes along with that is, what new technologies do we have to develop to create those new drugs? And so one example of that is our work on base modified TNA aptamers, which are kind of conceptualized here on the right side of the slide. So these are nucleic acid molecules that have a completely unnatural backbone structure, so they're biologically stable, but they also have side chains on the nucleobases that enhance their binding capacity. So these molecules in many ways have kind of the best of all properties. Okay, so they have the solubility and the flexibility of a nucleic acid polymer. They have the chemical diversity of a protein, and they have the biological stability of a synthetic polymer. And so I'm going to tell you a little bit about some of the things we're doing with our TNA aptamers, but before I get to that, I wanted to tell you about two other projects that are going on in the lab. And so the first one has to do with our work on gene silencing, and the second one, our work on diagnostics. And so this is sort of like the warm-up act for the main attraction. So in the area of gene silencing, we're working on a very specific problem, which is allele-specific gene silencing. And this idea is kind of conceptualized here in this slide. And so despite really impressive growth in the area of gene editing technologies, there still is a pressing need for therapeutic agents that can recognize a single point mutation in an mRNA strand and cleave that strand at that location to prevent the expression of a disease-associated protein. Okay, and this turns out to be a very tricky thing to do. So it's difficult for conventional gene silencing technologies to do this, like antisense and siRNA therapeutics. That's because these technologies are based on hybridization. And it's difficult to distinguish a perfectly hybridized duplex from a duplex that contains just a single base mismatch. And so as a consequence, these technologies, although very good, and there's many FDA-approved drugs for these technologies, they have a difficult time solving this problem. So what they tend to do is they tend to cleave both strands, the healthy strand and the disease-associated strand. So our approach to this is to use a different type of technology. This is the technology referred to as DNA science, or catalytic DNA molecules that are created in the laboratory by molecular evolution. And these molecules have two domains. They've got a substrate binding domain, which is shown here as this black bar. Okay, this domain can be engineered to recognize essentially any messenger RNA sequence using well-established Watson-Crick base pairs. And they have a second domain, which is the catalytic domain, that's kind of shown right here. This is a very specific sequence that can recognize a metal ion, it's typically magnesium ions, fold itself into a shape to cleave a specific dinucleotide junction. So the sequence shown here is a DNA enzyme referred to as 1023. It's perhaps the most well-studied of all DNA enzymes in the literature. It's highly specific for cleaving GU dinucleotide junction.
What are the superscripts mean, John?
Oh, those are the numbers in the
Those going
links,
around
in
the
the
site.
links as you go around the category. I'll get to those in a second. Okay, so in this project, we tend to use KRAS as a model system for an undruggable target that's caused by a single point mutation. And KRAS is, of course, one of the master regulators of cell division, and it turns out that in a large preponderance of cancers that you have these activator mutations that arise in the P loop of the GTP binding domain. And these effectively inactivate the catalase domain and prevent the hydrolysis of guanosine triphosphate to the diphosphate, which leads to cellular proliferation. Okay, and so what we would like to do is be able to recognize specifically the mutant strands by targeting that GU dinucleotide substrate and then leaving the wild type strand intact. All right, so our approach, although somewhat unique to this problem, faces a number of challenges. First of all, these are DNA molecules, which means that they're not going to be very stable inside of cells. They're going to struggle with biological stability. The other problem is that they're evolved in the laboratory to recognize magnesium ions, and they tend to require high concentrations of magnesium. So typically magnesium levels, say 5 millimole or higher, which is vastly greater than what you would find inside a cell. So if they're in a cell, they're going to have low cellular activity. And the other problem is that these substrate binding arms are also now substrates for the enzyme RNase H. And if RNase H recognizes them, it's going to trigger an antisense gene silencing mechanism. So our approach to overcoming this problem was to use chemical evolution. Okay, this is kind of an iterative design build kind of test process. And it uses our knowledge of nucleic acid chemistry to identify chemical modifications that we can insert into various aspects of these DNA molecules to overcome some of these challenges. And so what we did in this project was we started with the catalytic loop, and we walked the catalytic loop several times with different types of nucleic acid chemistry to identify positions where we could make chemical substitutions that would, for example, increase their activity, and also identify positions that are highly conserved where you can't really make any changes. And after several times walking the loop, we then moved on to the binding arms. And again, you're making modifications that would increase the biostability, but also increase the activity by increasing the affinity of the reagent for the RNA molecule. But of course, not so much that you get product inhibition. So there's a sort of delicate balance there. And in the end, through sort of 45 cycles of this kind of design, we came up with a molecule that we refer to as variant 46, which we also have kind of nicknamed DZ46. And you can see there's lots of different chemical modifications in there. And so we sort of started with the binding arms, which are modified in kind of a symmetric approach. We just kind of move in red, this is a locked nucleic acid. And then as you move sequentially in, you have three chemical modifications that are each a 2' methoxy RNA analog. We have 2' methoxy RNA analogs at position 7 and 8 of the catalytic loop. And then at position 14, we have a 2' no RNA modification. Okay. And then we also have these little loops here. These are phosphorothioate linkages that precede the guanine residues at positions 1 and 14. So when you take this variant with these types of chemical modifications, you can now achieve a very high level of catalytic activity under physiological conditions, or at least conditions that are simulated to mimic physiological conditions at 37 degrees. And so in the presence of, say, 100-fold excess substrate, we now get about 65 catalytic turnovers in 30 minutes, which is quite a bit more than what you would see, for example, in the unmodified DNA that I showed here in blue. And we were gratified to see that these molecules actually work well inside cells. So this is an example where we were targeting an endogenous KRAS target in an adenocarcinoma cell line. And what we were looking at was knockdown of the mutant G12V allele in the presence of the wild type allele. So when you look at, you can see this plot, what you're looking at is the abundance of the messenger RNA for the G12V and the wild type in untreated cells versus cells that are treated with low and high concentrations or low and high dosing of the DNA enzymes. And you see a selective knockdown of the G12V allele while leaving the wild type untouched. So that's some of the work we're doing on gene silencing. We've made newer reagents that are also kind of going after different targets, but it gives you a flavor of kind of a unique approach to gene silencing. And so the next sort of short story is on diagnostics. So, during the COVID pandemic, there was a great push for new types of diagnostics. We wanted to try to create something new in this space, and we wanted to create something that was a protein-free approach, so it's an approach that used purely nucleic acids. And we wanted to build from our work on DNA cells. And so the idea was we would build a multi-component DNA enzyme that would work as a sensor. So the way this was created was taking our original DNA enzyme, cutting it to create a split enzyme core, adding binding arms to the left and the right side that would then recognize the viral RNA target or analyte, and then when it's fully assembled on the viral mRNA analyte, you would have catalytically active DNA enzyme that could then cleave a substrate. And the substrate was designed as a reporter. We have a lot of different ways we design these types of reporters, but the most common one we use is a quenched fluorescent reporter. And so as long as this is bound, you get signal amplification in the presence of increased fluorescence through multiple thermal catalysis of that substrate. One other key aspect was, as we built this, we also introduced chemical modifications that increased the performance of this particular DNA enzyme. And then, for example, we reduce the limit of detection. Okay, so we call it a technology revealer. It stands for RNA encoded viral nucleic acid analyte reporter, which is a mouthful. We just say revealer. There have been a lot of different evolutionary sort of steps with this technology, but it originally started out as sort of a binary approach. Does the patient have COVID, kind of yes or no kind of result? And that was kind of our first paper in this field. And then we kind of moved on. We went from saying, okay, do you have COVID to can we do genotype? So for example, during the course of the pandemic, the virus was evolving, you had different strains, and some of these strains were strains we should be concerned about. They're called variants of concern. So could we genotype those concerns? And we had a system that allowed us to do that. And then we moved on to not only can you genotype it, but can you quantify the viral load? So we had a way to identify genotype and quantify. And this is kind of a cartoon image of our latest version, which is turning this technology into a diagnostic panel for lower respiratory infections. So the idea here is that a patient would present themselves to a doctor would diagnose them as having a lower respiratory infection, but of course then question which virus is affecting them. And so the idea is we would have a very, very fast, very inexpensive assay to do that. So you would collect patient nasopharyngeal swab, you would do isothermal amplification, and then you would treat that sample with our different sensors, each of which is specific to the different viruses, and then you could identify it that way. So that work is now recently been submitted for publication. So just to give you an idea of how we do the genotyping assay, to set this up, we had to create a competition assay. And so we basically had two sensors that would compete against each other. And then they were essentially trying to distinguish a single point mutation in the viral mRNA. So for the viral genome. And we had chemical modifications that would allow us to discriminate that base modification very nicely. And so these two sensors would compete, we'd have a sample, and it would come in and say, is this a variant of concern that you're interested in, or is it a wild type sample, or is it a healthy patient? And so you have a sample, and let's say it turned out to be a wild type virus. So both these sensors would be treated with that sample. And if it was a healthy or a wild type of virus, you'd have a large sort of high FAM signal, and you'd have a low hex signal. Okay. If it happened to be the variant of concern that you were testing for, you'd have a high hex signal, a low FAM signal, and if it was a healthy patient, it would be low in kind of both cases there. And so, this worked out really well. We developed sensors for kind of all the major variants of concern that were available at that time. We had clinical samples that we obtained from UC Medical Center. In this case, there were 34 clinical samples, 3 of which were from healthy patients, so patients that were in the hospital for a procedure but did not have COVID. The other 31 were from COVID patients, typically determined through a PCR assay. And so with each sample, you would treat it with the 5 different sensors in this competitive format, and you would identify which variant it was. It was very gratifying to see that the data we collected from patients treated at UC Medical Center really matched very closely with national profiling studies. And so what you're seeing here in the pie charts is really samples that were collected in early, mid, and late 2021. So you can see the viral progression, the emergence of epsilon, the emergence of delta variant, and eventually the emergence of the Omicron variant. And that matches, again, very nicely with national surveillance efforts. So when you look at kind of the data below, shown here is kind of a heat map. If you see the first patients that we tested, it's all blue. It was negative for these variants of concern. But we know it's a wild type because at the same time, we have 5 positive results for the wild type virus. And then in some cases, you have 2 positive results. And that's just because the beta variant coincidentally also shared this point mutation with Omicron. Okay. But of course, we knew it wasn't the beta variant because the beta variant never entered the United States. Okay. So then the third kind of installment of this technology was being able to quantify that. This was done in a format, a digital droplet format we call a digital droplet revealer. So it's very similar to, say, digital droplet PCR. Okay, so the idea here is there's no sample pre-amplification. You just take the viral sample that you denature the virus, you add the sensor, and then you encapsulate them in a microfluidic droplet. I'm going to tell you about this droplet technology in a little while. So you create these microfluidic droplets, you incubate them, you deposit them on a slide, you then image them with a microscope. There's tens of thousands of droplets. And in this case, we were using an AI software that would allow us to quantify the level of fluorescence in these samples. It's freely available on the internet. And then from there, we could quantify, based on linear calibration, the actual micro load. So this technology was made possible through kind of 2 innovations. One was multiplexing. So instead of using 1 sensor, we're now using 5 sensors and volume compression. So instead of doing this in bulk volume, we're now doing it inside microfluidic compartments. So you can imagine that most of these compartments are empty, but the ones that have your analyte in them now have that analyte pointed at a much higher concentration because of the boundary of the droplet. And this is, again, clinical data collected from samples at UC Medical Center. In this case, there are 20 samples, 2 of which were healthy patients. The other 18 had COVID. And you see a very nice linear correlation between quantitative real-time PCR data and digital droplet revealer data. And this was able to positively identify all these samples except one, which was sample 18. And it turned out that sample had like a CT level of like 35. So that's probably about the limit of detection for our technology. But that's also a patient that is most likely healthy because viral load is so low. Okay, so this kind of brings us up to therapeutic aptamers. I think most people in this audience are probably pretty familiar with the fact that human monoclonal antibodies have been an enormous success as therapeutics. They're a highly successful modality. If you look at the list of blockbuster drugs every single year, about half or at least half are human monoclonal antibodies. And of course, to be a blockbuster drug, you have to have annual sales of at least $1 billion per year. And so a hugely successful modality that's used to treat really a wide range of diseases. And these have some enormous advantages, right? So they have high target engagement, and they have long circulation time because of their large size. Okay, but they also have some challenges. So they struggle with, for example, tissue penetration. It's one of the big problems right now. They start to struggle sometimes with production because they're a biologic, which means they have to be produced in cells. Sometimes you get unexpected immunogenicity, and they still struggle with site-specific conjugation, which can be tricky if you want to put a molecule at a very specific place or on a very specific atom. Okay. So as a consequence, there has been a lot of discussion about what other technologies could be used that could not replace antibodies, but could mimic the success of antibodies and perhaps overcome some of these challenges. And one of those topics is aptamers. And you can see right off the bat that aptamers are much smaller than antibodies, but if you zoom in on them, they actually share many of the same physical attributes. So it's a single chain polymer that can fold into shape, that can bind to a target. If you look at the surface area that an aptamer covers, you compare it to a human monoclonal antibody, that surface area tends to be very, very similar. And of course, when they're properly developed, they have very high affinities and they can achieve very high selectivity. And they have some advantages, right? So they're generally considered to be cheaper to discover because they're not produced in animals. And they're generally considered cheaper to produce because they're not produced in cells. And they're often looked at as easier to scale and modify because they're generated by chemical synthesis. But of course, aptamer technology has been around a long time, and it also has a lot of challenges. And some of the same challenges I talked about previously, like biostability. So most of the aptamers that are in the literature are either DNA or RNA molecules, and they will never be drugs. Okay, in some cases they can be extensively modified to become a drug, but that's a tremendous effort, often with uncertain outcomes. And they struggle with effectiveness, which means they don't always bind as well as you might think, and also with throughput because they're historically very difficult to discover, right? It usually takes many, many rounds of evolution to find one aptamer that works really well, and then many, many rounds of directed evolution to get it to work better. So it's a significant effort to do this. But despite these challenges, there are at least 2 examples of aptamers that have received FDA approval. However, these aptamers are limited to ocular diseases. And this is due to the low abundance of nucleases in the vitreous humor of the eye. Okay? So if you want to create aptamers that are going to be more like systemic reagents that you could administer throughout the body, you're going to have to solve this biological stability. Because that's the main roadblock to doing that. And so one way to do that is to explore new regions of chemical state. And so the approach we've taken is to look at this artificial genetic polymer called Threomers nucleic acid. The chemical structure is shown here. So Threomers nucleic acid, abbreviated TNA, was originally developed by Albert Schiller during this period of time where he was trying to answer this interesting question about why did nature select or choose RNA as the molecular basis of life's genetics. And his approach was to take a chemical approach and say, you know what? RNA is an aldose sugar, and there are many aldose sugars. Why did nature choose a different aldose sugar than genetic polymers? And so, through an incredible effort, he built a series of genetic polymers, one of which was TNA, and just asked basic questions. You know, do they base pair themselves? Do they base pair with DNA and RNA? And things of that nature. So when I first read that paper, I was hugely fascinated by this work, and I've been interested, of course, ever since. Now, DNA uses the same genetic letters as DNA, but of course, it has this really unusual backbone structure that's highlighted here. And in fact, it has this unusual 2', 3' phosphodiester linkages, which is very different than, say, DNA and RNA, which are 3', 5' phosphodiester linkages. And if you actually count the atoms in the backbone repeat unit, what you see is there's 1 atom less in DNA than there are in DNA and in RNA. But despite these differences in the backbone, TNA turns out to be incredibly interesting. So one reason we like TNA is that it's incredibly biologically stable. Okay? We have yet to discover an enzyme that would degrade TNA. Okay? This work was originally done by Michelle Culbertson, who's actually an undergraduate student here at ASU. And so if you see names in the lower left-hand corner of my slide, those are ASU students that were here when my lab was here that were working on these projects. And this work has been repeated many times in many laboratories around the world, and in pharmaceutical companies, it's all the same result, which is that we can't find necessarily an enzyme that would degrade TNA. So it's incredibly biologically stable, which makes it a nice analog to work with. Now, the other surprising thing about TNA is that it base pairs very well with itself, but also with DNA and RNA. Okay? And so what you're looking at here is going to be the NMR structure of a TNA duplex that was solved by the Eschenmoser laboratory. And then on the right is a heteroduplex of TNA base paired with DNA from a crystal structure in my lab that was solved in complex with the polymerase. So it's very effective base pairing with itself and with DNA and with RNA. But there is a roadblock. So if you want to create TNA aptamers, you have to overcome a series of challenging problems. And so the first one is really a chemistry problem, it's the substrate problem. Okay, TNA building blocks are not commercially available. Okay, so if you want to make these, you have to generate them yourselves, and you have to generate them on scales that are large enough that you produce enough material that you can actually work with in the lab. So my lab spent years developing the chemistry, synthesizing these molecules initially, developing chemistry to synthesize the nucleosides on the multi-gram scale, say 20-gram scale, and then developing the chemistry to synthesize the phosphoramidites on the hundreds of milligrams to say 1 gram scale, and then also the phosphoramidites, which are the building blocks for solid phase synthesis. So we have some new chemistry coming out of triphosphates. I'm not going to talk about today, but it's kind of exciting. I think it's very generalizable that could be applied to other analogs. So this is a big problem, but it's a problem that can be solved with chemistry. This other problem over here is a much bigger problem, okay, which is the polymerase. So natural polymerases don't recognize TA. Okay? So if you want to create aptamers in the laboratory, you're going to need a polymerase. And the first step of your project is to invent a polymerase. So when the first step of your project is to discover the enzyme that you need to do the thing you need to do, you know it's going to be a difficult project. So to do that, we had to invent our own technologies. So we invented a way to evolve polymerases in uniform water oil emulsions. Okay? These are microfluidic droplets, and this is a single cell molecular evolution approach. And the way it works is that we design our libraries at the DNA level. We transform an E. coli, we grow express E. coli, so each E. coli is essentially expressing a different polymerase variant. We encapsulate E. coli in water oil droplets. We lyse the E. coli. This releases the plasmid and the enzyme into this fermented solution. Now you have this genotype-phenotype relationship. If the polymerase does what you want, you're going to isolate that droplet, and then we can determine the sequence of the enzyme by sequencing. So that's kind of the technology. It has a number of pretty big advantages, but the two biggest advantages is throughput, so we can screen millions of variants in a day, and it also has enormous economy of scale. This is done on a scale that's about 1 million times smaller than, say, conventional liquid handling robots. And that's important if you're making your own hypothesis. So this is sort of what our evolutionary cycle looks like for polymerases. We refer to this as drops, which is kind of a cute name for the droplets, but it stands for droplet-based optical polymerase sorting. We take our library of polymerases that are expressed in E. coli, we encapsulate them in water oil droplets, we encapsulate them with a fluorescent sensor. Okay, so this is really just a self-priming template that has a fluorophore on the end. There's another DNA molecule that's a quencher. So as long as they're in close proximity, there's a quench droplet. Okay, then we heat these populations with droplets to lyse the E. coli. So we always work with thermophilic polymerases. Okay, and then we incubate them for periods of time. And the incubation time is generally longer than you expect, and that's because there's an enormous dilution effect when you go from the volume of a single bacteria cell to the volume of a compartment. So you have to give it enough time to react. Then we bring it back to room temperature. If the enzyme is able to extend the primer all the way up to the length, you have a fluorescent droplet. Most of the droplets are not going to be fluorescent, some will. Now we have to isolate those fluorescent droplets. To do that, we use a second microfluidic device that allows droplet sorting. This is referred to as a FAS device or a FAS instrument, so it's a fluorescent activated droplet sorting, very similar to FAX sorting that many of you might do. So you have droplets that are flowing through a channel. They're going to pass through a laser, and this is laser-induced fluorescence. And so if the fluorescent excitation spectrum matches user-defined parameters in terms of height, width, resonance time, and declares it a fluorescent droplet, then on a microsecond time scale, the computer will trigger an electrode that's usually down here that fires a series of pulses that then allows the droplet to change electrophoric mobility. Hit this divider and kind of roll it in. Otherwise, they tend to be moving forward. There's an extra stream of liquid here pushing them up. There is this sort of natural force keeping them going through. Once we collect our sorted droplets, we can isolate the DNA. We can take it through another cycle of evolution, or we can sequence it to get it working. This is an example of what one of our microfluidic chips looks like on the left. It's mounted on an inverted microscope with a high-speed camera that allows us to visualize the formation of our droplets. So this is a flow focusing device. And so you've got your aqueous solutions coming to the left, your oils and surfactants from the top and the bottom, and you have a very nice and very stable dripping routine. And all the droplets are roughly uniform in size. So this makes droplets at a rate of about 35 kilohertz, so that's about more than 100 million droplets per hour. Now, most of our droplets are empty, so in order to ensure it's only one bacteria per cell, you have to use the percent distribution. We usually use it with about a 10% occupancy level, so about 90% of our droplets are empty, but the ones that do have a bacteria generally have only one bacteria. And then once we have this, we collect them, we incubate them, we bring them back to room temperature, and then we can sort them on another device. This is kind of a mock sorting video, and so the dark ones have died, they're empty. The clear droplets are fluorescent. You see there's our electrode, you see them slightly getting tighter. It's not a huge effect, but it's enough to bring the droplet down to hit kind of the device and roll into the bin. So we can sort it at a rate of about 1,000 droplets per second. So when we're doing big evolutionary experiments, we're starting for 1 to 2 days. You can see this droplet here kind of getting caught in the act of being tugged down. Right? So, so this works. We've evolved a lot of TNA polymerases this way. Most of them were not very good. Okay? And so what you're looking at here is a theoretical fitness landscape, if you will. And most of the polymerases that we've evolved are in this orange region over here. A lot of these polymerases came through deep mutational scanning experiments, where we systematically scan the gene one at a time and look at the other 19 amino acids at that position. Then you find one that works and you try to recombine it with another one that works over here. That turns to be very difficult because of effects like epistasis, but it works. And so this polymerase here called CODRAS-G, that's our previous best. And it was discovered by individually discovering the RDS and GNA mutations, which are otherwise unnatural COD wild type backbone. And so, these things work, but they're very slow and they tend to be more error prone. What we really wanted to do is be up here, right, in this region, on a high key with something that's really fast and something that is very accurate. It's something that's more reminiscent of what you would find in nature. Now, this is one of the big challenges, not only in polymerase engineering, but just enzyme engineering in general. Okay, so the way we decided to get there is to explore new regions of the fitness landscape. The way we did that is to turn to homologous recombination. This is a technique that's been used a lot in the literature for different applications, but we decided to try it out. And the way we did is we used 4 archaeal DNA polymerases. These are all isolated from extremophiles that live in like thermal vents and grains. Okay, we chemically synthesized them by G-block assembly. We designed all the sequences at the DNA level. We added our RSGA mutations that we knew kind of helped DNA synthesis. And then these bars signify regions that are 100% identical between the 4 variants. And you need those regions that are very important. And so then we did homologous recombination by PCR. So we used those regions to design primers, and we individually amplify each of these pieces out. So this is a disassembly approach followed by a reassembly approach. So you disassemble everything. So in this case, it's 44 PCR reactions. Put it all together, amplify it back up, and now you've got a homologous recombination library. But because it was created by PCR, you have more diversity than you initially thought. So the diversity, you get all of the expected recombination events, which in this case was formulated, but you also get novel recombination events. And this is due to template switching that might occur during PCR. And then you also get kind of random point mutation that sometimes come in. And so you get a very rich library. Most of these enzymes do not have stop codons. Okay, so this is very different than, say, error-prone PCR methods, where they tend to generate a lot of stop codons in the methodology. Okay, so using this library, we did a directed evolution experiment where we took our library through 10 rounds of evolution. This work was recently published in Nature Catalysis. It's available today for the first time, which is kind of coincidental with this talk. So for this library, we initially did 5 rounds of just sort of enrichment for things that are TNA activity. So it's negative selection, kind of get rid of things that weren't very active. Then we did 5 additional rounds of directed evolution. What you see below, in parentheses, the number of clones that we screened at that round of the selection to identify the best variants, which is shown above, in this case 5-0. That was the 270th clone from round 5. That was then derivatized and carried forward. So here we mutate the thumb, around 7, again, screen, identify, mutate, round 8, and eventually getting to round 10. Okay. And during the course, we did extensive biochemical and structural characterization. So all of those major intermediates, we saw all their crystal structures, both as the binary structure, but also as the closed ternary structure. We measured the kinetics, we measured the fidelity. So some of that data is shown here. So this is the kind of kinetic data. And so what you see is going from where we were before to a very slow enzyme up to 1092, now an enzyme that has a rate of about 70 nucleotides per minute, so a little faster than 1 per second. That is within 5 to 200 fold of the native activity of the 4 polymerases that we use for life. So that was a significant improvement, and for the first time getting close to a native. But for an extant system or an artificial genetic system like this, that was a major improvement. But we also very gratified to see that as we improve metallic activity, we also increased the fidelity of the So it has a fidelity of about 99.9%, which means it makes about 1 or 2 mistakes out of about every 1,000 nucleotide incorporation events. Okay. Now, what are these? How many mutations are there? What are they doing? Well, it turns out there's 51 mutations relative to the wild type sequence. Not all of those came through the selection. 2 are in gray here. These are exonuclease silencing mutations that were there by design. In blue, these are RSGA mutations. In gold are the mutations that came from the initial part of homologous recombination and evolution. And in red are the ones that were isolated from direct evolution. But when you look at this, what you see is the mutation is treated all over the protein surface, which is really interesting, actually. And so then you begin to ask the question, what are these mutations doing? So the best way to do this is, of course, solve the pressure. So we solved it in this catalytically active closed ternary complex, and we overlaid it against the wild type structure. So the wild type structure here is in gray. The 1092 structure is in pink. And what we saw was major structural rearrangements. You see this very nicely if you zoom in kind of this box shown here on the upper right. You see this alpha helix that shifts by as much as 7 angstroms. You also see major rearrangements of these loop regions, in different aspects of the thumb. And likewise, down here in the palm, we see major structural changes. This actually included an insertion of an amino acid that shifted this alpha helix down. Okay, and the thumb, of course, is very important for translocation of the enzyme. And the palm is very important for catalysis. That's where the catalytic aspartate residues reside. And so the idea, or the picture that's beginning to emerge, was that these major structural changes were really fine-tuning the enzyme active site, or the geometry of something in the enzyme active site. And so if you look in on that, what we're seeing is that the active site is becoming much more wild. And so what we're looking at here is a zoom in of the wild type DNA polymerase. So this is a DNA triphosphate. These are 2 of the 3 catalytic aspartate residues, and then these are the divalproenyl ions. And if you compare that to, say, the polymerase that was isolated from around 5, 8, and around 10, what you're seeing is stepwise improvements in the geometry. And you see that especially at this catalytic aspartate 404, where you go from green to blue to purple. So green is far out, it's kind of facing the wrong direction. By the time you get around 8, it readjusts, and by the time you get around 10, it now closely approximates the aspartate that you see in the blood. So that was really exciting for us to see, and it kind of changes our views on what are the determinants of substrate specificity. We also saw these changes in the orientation of the substrate in the active. So again, this is the wild type on the left. And if you just focus in on, say, the distance between the sugar and the alpha phosphate, so where you make the phosphodiesterase bond, what you see is in the wild type structure is 3.7 angstroms. At round 5, it was a very distant 6.7 angstroms. By round 8, it was down to 4.3. And then when you get to round 10, it's now 3.9, which closely approximates the wild type, and also sort of explains a lot of catalytic activity we're seeing this. Okay, so I think these observations together suggest that polymerase specificity is governed by global structural topology rather than a precise set of active site mutations, which is very different than the historical view of how the re-engineered polymerases. This is a result that we also see in work I'm not going to tell you about, where we evolved an RNA polymerase that can synthesize a broad range of RNA analogs for vaccine development. So we've seen this now in kind of 2 different evolutionary studies. So what do you do with an enzyme that didn't exist before? Okay? But what we want to do is we want to create an on-demand approach to therapeutic aptamers for preclinical drug discovery. Okay? So this is a vision we've had for many years now, but the technology to do this didn't really exist until the last year or so. So now we have kind of all the tools in place to build this pipeline. We have this wonderful new enzyme that allows us to build lots of different types of libraries, library compositions, even chemical modifications. We've now highly optimized target screening. So we can do one round of screening. It's no longer an evolutionary experiment. This is much more akin to what we refer to as a DEL approach or a DNA encoded library approach for small molecule discovery. And we then sequence. We now can do secondary screening in 96-well format for hits. This turns out this was not a trivial problem to solve. These are not DNA aptamers. You can't just call up IDT and say, hey, send me 96 DNA aptamers. They're going to ask you what's DNA, okay? So you have to develop your technology to do that. Then we use that technology to hit optimization. And now, through a lot of extensive chemistry, we can solid phase chemical synthesis, even on base modified things, and validate those aptamers. And then eventually getting out to functional studies. Okay, so how do we do this? Well, we do our selections in a sort of DNA encoded library approach. So we have our libraries, they're self-priming hairpins, so there's black regions and random region. We use our TNA polymerase and we extend it out with TNA. Now you've got kind of a hairpin structure that's very long. Okay, now we come in with a second DNA primer that's very long. We anneal in this region, okay, and we extend that out with the polymerase that has strand lengthening activity all the way in. So now you've got double-stranded DNA that's linked to the single-stranded TNA, and then we fold those TNA molecules. And then this allows you to create, again, that genotype-phenotype linkage that's required for molecular evolution. We can query this library against targets, things that bind. We can then photocleave off our DNA, recover the DNA, PCR amplify it, and sequence. So it's a very straightforward sequencing approach. One of the problems we had to solve along the way was how do you get aptamers that are more effective? And largely what we mean by that have the binding properties that are more reminiscent of an antibody. Okay, so one of the reasons why antibodies are so good is because they have slow off. So what you're looking here is kind of a hypothetical kinetic analysis of an aptamer protein binding interaction. So this is something you can measure with BLI or SPR. You have an on rate, and then when you take the ligand away, you have a slow off rate. And that's why antibodies work well. They bind their targets and stay bound. Aptamers most often, especially protein binders, and they have fast on rates, but then also fast off rates. So the question is, how do you turn an aptamer with fast off rate to one with a slow off rate? And the answer, of course, is to look at the nature. So if you look at the principal structures of antibody antigen complexes, peel them apart in what's called the open book view. What you see is if you look at the paratose surface, so the surface of the antibody that contacts the epitope, you see a large number of aromatic rings. This is not by accident. It turns out that almost all antibody antigen complexes have this. So this is one study that looked at 53 different structures. And you see that these aromatic amino acids like phenylalanine, tyrosine, tryptophan are about 5 times more abundant. Now, we're not the first to look at this. There's a lot of academic labs that have done this, a lot of companies that have done this. But we're really the first to do it on artificial genetic polymers. So the first example is a set of 10 analogs that we chemically synthesized as nucleoside triphosphates. So it's a uridine TNA nucleoside triphosphates that are modified at 5 positions, but C5 modified uridyl derivatives with all these different analogs. Each one was about 18 synthetic steps, so you're looking at about 180 synthetic steps to make these analogs. We've made somewhere in the 30s of these. We've also made C5 base modified cytidine analogs, linked for these two, and then most recently for tryptophan. So we have kind of an ensemble of analogs that we can work with. And gratifyingly, we see that when we do binding studies with these, we get very nice on rates and then very slow off rates. And this was an example that was done with just one type of modification on uracil. I think it's with the tryptophan. Okay. So a few years ago, quite a few years ago now, we published a review on aptamers. One of the things that became apparent, because we downloaded every single aptamer paper in literature, and we looked at, especially the discovery papers, is that aptamer papers that report the discovery of new aptamers tend to focus on the same 10 targets, number one, And number two, of the ones that they report a new target, they usually characterize only one sequence from the selection. And we all know that selections produce more than a sequence. So we wanted to look at lots and lots of sequences from the outputs of our screens. And we needed to come up with a way to do that. We decided to develop a technology that's really a hydrogel, we call hydrogel particle display. So these are basically magnetic beads that are coated with a polyacrylamide gel. And we make these in bulk solution, but they're remarkably uniform inside. And so the way this works is you've got your hydrogel particle that you make, and you cross-link a DNA primer in the matrix. So you purchase a DNA primer that has an acridite functional group on it, you add it in, and it gets cross-linked. These gel surfaces are very porous, so reagents can go in and out. So you can add a template, you can add your phosphates, you can synthesize your DNA aptamer, you can remove the template by changing the buffer conditions. You can heat these up and anneal and fold the aptamer, and then you can query the aptamer for activity in the ELISA-based assay. So this is a simple example where you have a biotelated target, you come in with a struct data and fluorophore, and then you just read these out by flow cytometry. You just flow these through flow cytometer and you look for shifts in fluorescence. So being able to do this in one bead allows you to do it in a nice well format. And so when we do these screens, we typically only order 96 sequences. We have to, we're a little careful on how we pick sequences and look for certain things. We synthesize them, of course, enzymatically, using their templates, strip away the templates, do an ELISA type assay to identify which, if any, of the hits in that set of 96. And when it works well, we typically get about 3 to 4 hits. Okay, so this is an example of a selection experiment that we did with one type of base modification, it's the tryptophan modification. And we did 6 different experiments in parallel. And in each case, it's only one round of screening. We sequenced, we identified the hits. These are hits with standard chemistry, with chemistry, cyclopropyl, diacetyl, phenylalanine, and tryptophan. And in this particular case, the tryptophan worked very well. Phenylalanine, maybe just a hint. But most of these, especially standard base, show no activity. And then you might expect for a single round of selection. But once you can do this, you can now say, well, tryptophan seems to work well with this target. Let's change the library a little bit. And you can do different library links. So in this case, you change the library length from 20 to 25, 30, 40. These two aptamers here are the ones I showed you the biolateral time before. We get a nice slow off rate. So even with short libraries, we get very nice aptamers. We can also do things like SAR studies. So this is the end mapping truncation analysis study, where you individually delete mutations, residues from the 3' end and from the 2' end. Remember, the 3' end of DNA is like the 5' end of DNA. Okay, and so you can truncate it down to what's the core region. You can do SAR studies like scanning genesis. You can swap bases. You can do medicinal chemistry by putting in analogs. So you can do a whole lot of things without ever having to purify the aptamer. And that's hugely important. So we can do these assays in like 1 to 2 days. If you wanted to make and purify 96 aptamers traditionally by gel electrophoresis, maybe the gel bag, you're looking at 6 months of work. Okay, so there's a great acceleration. We now have a system where we've taken 14 targets, at least through parts of these pipelines. All 14 have gone through primary screening and NGS analysis. The vast majority have gone through secondary screening and hit optimization. And 2 have gone through chemical synthesis and independent validation of a chemically synthesized product. These are the 14 targets that we've looked at thus far. They're very recognizable to many people here. The 3 that did work in blue, we believe did not work for a technical reason that they all share. And so we're going to go back and look at that. But generally they all work really well. So we can get aptamers from a single round. This is a chemically synthesized aptamer. It has modified cytidine and uracil residues. And you see it has very nice antibody-like characteristics, right? You've got a very nice KD, you've got a very nice slow off rate. And the data is very reminiscent of this that we're seeing. So typically it's kind of between, say, 25 and 500 picomolar binding. So for a single round of selection in a system that's totally unnatural, using an enzyme that doesn't exist in nature, that's a pretty big step forward. I think here what I'll do is I'll just stop and summarize some of the main points that I see. So the first one is that artificial genetic polymers, which we call exodays, have physicochemical properties that are desirable for many diagnostic and therapeutic applications. We saw that in some earlier work. I would say that polymerases play an oversized role in biotechnology and healthcare. And so if the past is any indication of the future, then technologies that can efficiently reprogram DNA polymerases for new types of activities have the potential to really uniquely drive new applications in biotechnology and medicine. And then lastly, that this on-demand approach to therapeutic aptamers could provide a new modality that could complement the success of therapeutic antibodies. And I want to obviously thank all the students in my lab, my collaborators, for the funding, and I'm happy to take any questions you might have. Thank you.
John, amazing. Your polymerase is incredible. I think the generic folding problem with pressure from the outside of the enzyme is something maybe to be expected. How good was AlphaFold compared with all the hard work you did?
I love that answer. I love that question. What do you think? What would you predict? No pun intended.
I honestly don't know, but AlphaFold does so well these days.
AlphaFold, I get this question every talk I do. AlphaFold is really good at well-structured proteins or proteins that bind very defined ligands. But polymerases are so dynamic, very difficult for them. I know they're working on that. Also, enzymes that bind substrates are difficult, especially DNA substrates. This is even more difficult because it's DNA and a triphosphate. So we specifically cataloged and very accurately analyzed the AlphaFold and the Rosetta Fold program to do this, and they were off by quite a bit. So it was nice to see that, and we may write a little comment here. But I think people that are in the niche know that. People that just read the news and reviews kind of thing think that the problem is 100%.
Thank you.
Yeah.
Is there any, so when you finally got your final polymerase that you're using for your TNAs, did you go back and search anywhere that anybody's ever sequenced the polymerase that's similar?
Not at all. Yeah, we've done it like last now.
Yeah, just nothing.
Very unique.
So it really is brand new.
Yeah. Also, 1092 was rare in round 10. Like we screened those twice because we weren't entirely sure. And all the activities were better than round 8, but only that 1092 was substantially better. That tells us it was pretty rare in the population. At that time, we kind of got lucky.
So we got to go to another planet in order to find
It might be out there, right? You know, let's go to Mars or something.
For your detection technology, how cost effective do you think you can get that to be able to be used in low-income countries?
Remarkably so. It's quite a bit cheaper than the comparable CRISPR approach. So there's two different kind of CRISPR approaches that are being used for that currently. And when you do this kind of the cost benefit analysis, it's definitely more than half the price of those technologies. So there is no protein expression, right? These are oligos you could literally buy for money. Yeah.
Great, thanks.
I have a question regarding to your gene silencing project. So, for example, they can be used to treat KRAS, wild mutants, colon cancer, for example. So, how do you, use strategy to deliver those DNA-based enzymes into cancer cells?
Yeah, no, that's a good question. So we are working with collaborators in industry that are modifying reagents with ligands go in, both in animals and also doing cell work. And that work is kind of ongoing. But the current thought on that is even as good as that molecule is, it's not going to be good enough as a therapeutic because there's too much DNA content in it already. We have improved that, but I think we've hit the limit. And so the new work is designed on creating the same type of reagent, but entirely out of TA. And then it would be very hardy, very biologically stable. Then you could modify it with various ligands and do like a galvanic person.
Okay, thank you. Very nice story. I just curious, your first part of story, you use the RNA for target RNA. So it's about 13 nucleotide to match, right? So the two RNA in your first part of story.
For gene silencing?
Yeah. So my question is that how do you detect the off target for those kind of design?
Right. So the most common design that we publish on is the 7+8 design. So it's 15 nucleotides that it recognizes. So one way to do it is to look at those specific 15 nucleotides and say, does anything remotely close to that show up in other genes in the cells or cell lines you're looking at, or even just in the human genome? And so that's one way to kind of parameterize it. We have in our first study looked at, I think, up to like 50 off-target or closely related targets, and the activity is very good. So these tend to be very, very selective, actually. Remarkably so, more than I thought they would be.
Okay, thank you.
Any other question? Oh, one more.
I have a question on DNA enzymes, like because your catalytical code is flexible, right? So will there be any issue in stoichiometry? Like let's say you have RNA substrate 1 micromolar and DNA enzyme 2 micromolar. So can two DNA enzyme binds there?
Oh, it's stoichiometry.
Yeah.
No. It's only one. There's now actually a crystal structure, no, an NMR structure of the 1023 DNA time. And the fold is now much better understood. And it can only accommodate one person. Yeah, so one DNA enzyme per mRNA target. The stoichiometry is one. And then the folded structure is now elucidated. So we're actually collaborating with that team on our chemically modified version to see, what additional conformational changes transpired, or do we see greater rigidity, for example, in the resulting one that was chemically modified.
Thank you.
Oh, one more.
Hi, very nice talk. So I'm just curious, in your lab or maybe some other group studied whether TNA induce any type of innate immune response? Is it known?
Not yet that we know
Okay.
of. We have had TNA oligos that are aptamers go into animals, and we have studied that, and there isn't anything that's immediately apparent. There's also been cell work done. And the TNA modifications themselves, we also have an active collaboration with Alnylam looking at TNA modifications in siRNA